Good day, everyone. I am Lyle Ijssel Guzman, and for my journal report, I will be discussing Xeroderma pigmentosum. The Xeroderma pigmentosum, or what we call XP, is a rare autosomal recessive genetic disease characterized by hypersensitivity to UV exposure associated with numerous skin abnormalities, ocular abnormalities, and a very high predisposition for developing skin cancers. XP results from mutations involved in nucleotide excision repair or NER pathway, leading to a defective repair of DNA damaged by ultraviolet or UV radiation. It is a very rare disease that affects only 1 out of every 250,000 people worldwide. Because of its rarity, XP has been referenced in pop culture many times because of its phenotypic characteristics. Clinical manifestations include exaggerated response to UV radiation. Short exposure to sunlight causes pronounced burning and blistering, as seen in the photo on the left. Freckle-like pigmentation of exposed areas upon long-term exposure can also be observed. Growth of a tumor in UV radiation-exposed areas can be observed on the skin, eyes, and mouth. Some XP patients are also diagnosed with progressive neurological degeneration. With its grave manifestations, life expectancy of XP patients are significantly lower than the general population. The median age of death of XP patients is 32 versus the 67 years old for general population. Meanwhile, XP patients with neurological degeneration has a lower median age of death at 29 years old. Cancer and neurological disorders are the top two causes of death in XP patients. Surprisingly, XP has been found in all continents and across all racial groups with very low occurrence rate. So what causes XP? XP results from mutations in any of the 8 genes involved in the repair of ultraviolet-induced photoproducts in DNA by the nucleotide excision repair or NER. Seen in the table below are the XP genes involved in the NER pathway. Later, we will discuss how each proteins perform in the damage repair. Let's first have a quick recall on how DNA acquires damage and what are the cell's mechanism to repair these damages. We can classify DNA damage into two types, endogenous and exogenous damage. Endogenous damage are caused by attacks by reactive oxygen species and replication errors. Meanwhile, exogenous damage are caused by external agents such as UV rays, ionizing radiation, and chemical agents. Cells respond to these damages by utilizing the following repair mechanisms: your direct reversal, single-stranded breaks, and double-stranded breaks. We have taken this up in Chem 240. For UV-induced damage, cells specifically use nucleotide excision repair pathway. When a person exposed to sunlight, UVA and UVB can pass through the skin and damage the DNA within cells. UV irradiation causes two major photoproducts in DNA, 6,4-pyrimidine pyrimidone or 6,4-PPs, and cyclobutane pyrimidine dimers or CPD. The lesions, when not fully corrected, causes mutations in the DNA which may influence cellular death, aging, and carcinogenesis. The cells repair these lesions through the NER pathway. This pathway can proceed through either of the two repair mechanisms, the global genome repair or GGR and transcription coupled repair or TCR. TCR repair lesions located on transcribed strand of active genes. Meanwhile, GGR detects and removes lesions throughout the whole genome and has specific affinity for injured sites. Regardless of which pathway the cells undergo, NER can be summarized into 3 steps: recognition of damage, excision of damage by endonucleases, and synthesis of removed DNA region. For UV radiation-induced damage, we will focus on the global genome repair. Presented in the slide is a general overview of global genome repair mechanism. For the first step, XPCRAD23B initiates the GGR-NER by recognition of the damage. XPC binds to the ssDNA opposite of the lesion, and the recognition is sustained by the uvDNA damage binding protein or UBDDD2. A transcription factor TFIIH is recruited to the lesion site using the energy provided by the ATPase activity of XPB. XPB and XPD are both subunits of TFIIH and are ATP-dependent helicases. Your XPB facilitates the binding of XPD to 5' of the damaged site, and XPD scans the strand and stalls upon encountering a lesion. XPA and RPA are recruited to form the pre-incision complex or PIC. PIC is stabilized by endonuclease XPG. Your XPC leaves the complex upon arrival of XPG to the PIC. The single-stranded DNA binding protein RPA meanwhile binds to the undamaged strand while the transcription factor opens the duplex. Your XPA recruits structure-specific endonuclease XPF and incision on both sides of lesion takes place. Your XPF cuts 5' while XPG cuts 3' of the lesion. This results in elimination of 22 to 32 nucleotides. Lastly, the DNA replication will be performed by polymerase and ligation by DNA ligase. The table shown summarizes the role of each XP complementation group and the corresponding phenotypes associated with mutations in each XP genes. Due to variety of the disease, listing every mutations will be lengthy. Different clinical manifestations are observed for each XP subtype, with XPA and XPB mutations showing severe neurological symptoms. Meanwhile, mutations in XPC, XPD, and XPE are the most common genetic alteration observed in European and Japanese patients. Currently, there is no cure for XP because DNA damage is cumulative and irreversible. The best course of action for patients include early diagnosis and protective measures to prevent occurrence of skin cancers and prolong their life expectancy. Persons with XP must avoid exposure to any sources of UV light including sunlight, fluorescent, halogen, and mercury vapor lights. UV-absorbing films and filters can be placed over windows and fluorescent or halogen lamps. Sun protection can be achieved by wearing protective clothing and UV-absorbing eyeglasses. Vitamin D deficiency upon sun protection can be alleviated by vitamin D supplementation. For medical care, patients can be prescribed with oral retinoids or vitamin A derivatives or topical therapy with 5-fluorouracil. Potential therapies also include the use of bacteriophage 4, endonucleases, and photolyase. Surgical management, meanwhile, includes complete removal of skin cancers and dermatome shaving or abrasion. Regular visits should also be paid to dermatologists for early detection and treatment. Frequent eye examinations are recommended as well as neurological evaluation. Patients should also receive follow-up care every 3 months, focusing on educating the patient about effective sun protection and early recognition of skin cancer. Genetic consultations should also be made for families at risk. Lastly, gene therapy can also be an approach in treating the disease by targeting the mutation in the gene. Advances have been made in the field of gene therapy. Several studies include targeted gene correction of XPC gene, the most common XP subtype. Targeted gene therapy of XPC gene is based on the repair of a DNA double-strand break, DSB, by homologous recombination. Mutation in the XPC gene include deletion of TG residues at the 9th exon causing frame shift and premature stop codon. Ultimately, this results in a truncated XPC gene. The strategy for gene therapy involves an endonuclease-induced targeting approach with the use of an engineered endonuclease, either meganuclease or transcription activator like effector nuclease or TALENs. For this approach, an engineered endonuclease induces a DSB at a given locus. The double-strand break stimulates the homologous recombination process in the locus. In the presence of a DNA repair matrix, the recombination may result into insertion of the new DNA sequence of correction to the mutation. Meganucleases are naturally occurring endonucleases which have the capacity to recognize and cut large DNA sequences from 12 up to 40 base pairs. The structure of common meganucleases is derived from a natural homing endonuclease, the crystal structure of which is shown here. Because of its high sequence specificity, meganucleases are widely used in targeted gene correction. However, its efficiency is limited by epigenetic modifications. In a study by Dupuy et al., they engineered a meganuclease targeting a locus near the founder mutation in XPC genes, which contains two methylated cytosines. Since meganucleases are sensitive to DNA methylation, cells are treated with demethylating agent before subjecting to the engineered endonuclease. An increase in the targeted mutagenesis is shown for cells treated with the methylating agent. Meanwhile, sequencing of the homogeneous genome targeting PCR products showed gene correction at the target locus as confirmed by the insertion of TG nucleotide in the gene. This is the mutated gene, and this is the corrected gene. And this is your control. As you can see here, the TG deletion is successfully reversed. On the other hand, TLNs are produced by fusion of the restriction fork 1 endonuclease domain with the high specificity DNA binding domains from Xanthomonas bacteria. The DNA binding domains consist of a highly conserved 33 to 34 amino acids with divergent 12 and 13th amino acid. These two FOC1 molecules, when they bind to their recognition site, they dimerize to form a functional endonuclease which cleave at the spacer sequence. In contrast to the meganucleases, TLNs can be modified to be insensitive to methylation, and thus a study by Dupuy et al. Makes use of a TLN that is engineered to target the XPC gene while being insensitive to methylation. Shown here are the two graphs from the study. The first graph measures the survival rate of cells exposed to UV radiation. These cells consist of the proficient for XPC cell line, which is the MRC5 or the positive control, and then for the parental cell line, characterized with its TG deletion, the XP4PA, while the corrected clones are the corrected clones 3 to 7. The green bar denotes unexposed to UV radiation, and black bar denotes cells exposed to UV radiation. As you can see there, the parental cell line has little to no survival upon radiation with UV. Meanwhile, your clones 3 to 7, which are treated with TLNs, have a significantly higher survival rate. For the second graph, the kinetics of 6,4-pp lesion repair after irradiation is also measured. The closed circle and triangle denotes the corrected clones, while the open square and circle denotes the uncorrected clone and the parental cell line respectively. So for the closed circle and triangle, you can see there an abrupt decrease in the amount of cells containing the lesion, meaning that there is a repair mechanism in the cell that allows it to remove the lesion, and that is due to the treatment with TLNs. Meanwhile, little to no change in the number of cells containing the lesion is observed for uncorrected clones and the parental cell line. These two graphs confirm the efficiency of TLNs in treating XPC gene mutations through targeted gene therapy. To summarize everything, XP is a rare autosomal recessive genetic disease characterized by hypersensitivity to sunlight, skin and ocular abnormalities, and an increased risk of acquiring cancers. Mutations in any of the XP genes involved in the nucleotide excision repair pathway results in the development of the disease. Subtypes of the disease depend on the complementary group mutated. There are a total of 8 XP subtypes. Treatment includes protective measures from UV radiation, oral and topical medication, monthly consultations, and follow-up care. Targeted gene therapy with meganucleases and TALENs provides a promising approach in alleviating the symptoms of the disease and improving the life of XP patients. That is all. Thank you, everyone.